Alright, hey everybody. So today we're me talking about guanylate cyclase 2D and a little bit about its role in vision. So what is guanylate cyclase 2D? So it is a type of gene that aids in the making of a protein called retinol guanylyl cyclase 1, also known as RET-GC1. So in this class of protein or enzyme, some will call it, is a type of membrane bound class. So it is essential for basically visual recovery after bright light. So basically, when you look at a bright source of light or like the sun and you look back to the ground, there's this like, you know, this blur in front, and that's basically the guanylate cyclase 2D that's working inside your retina. So it is, a crucial role in CGMP production in the retina. And then we'll learn a little bit more about what exactly does CGMP do in this cascade. So a quick fun fact is that mutations in the GUCY2D causes early childhood blindness. So where and when is guanylate cyclase 2D expressed? So it is primarily expressed on the outside of your photoreceptors, so the cones. So there's two types of photoreceptors, cones and rods, and cones is what helps you see color in the retina. So it is involved in the process called phototransduction. So once light hits your retina, they want to go back to the resting stage. So guanylate cyclase 2D is involved in the chemical reaction that kind of reopens the channels that returns the photoreceptors so that cascade can happen again. So basically, guanylate cyclase 2D will code for RET-GC1, which in turn converts GTP to CGMP, as shown here. So it has a pretty crucial role in biochemically and some substrate and ligand interactions as well. So the substrate for RET-GC1, so I'm be referring to RET-GC1 as the protein product, or the product that was encoded by the guanylate cyclase 2D. So that is GTP. So the end goal is for GTP to be converted into CGMP, which is the molecule that is needed to regulate, the visual pathway we talked about. So this activity is heavily regulated by calcium ions. So as we learned in class last week about, biomolecule transports, or, just membranes in general. So basically these ions will signal, to close or open a membrane. So in this case, a low calcium concentration will activate the RET-GC1 to produce CGMP, so it can open up that channel again for calcium ions to flush in. And while on the other hand, high calcium levels will inhibit it. So this ultimately is kind of the regulatory factors of how CGMP is getting inside of your cells. So basically, how it works is RET-GC1, or guanylate cyclase 2D. It binds GTP in its catalytic domain, also known as a active site. That's why we consider it a enzyme. So once the GTP binds to the guanylate cyclase 2D, the enzyme becomes activated, and thus it converts GTP into CGMP. And this interaction contains a lot of weak interactions we talked about class, because that's how we are going to release that final product of CGMP, because if it binds too tight, then it's not going to release it. So in that active site, there's also lysine and aspartate amino acids that helps position the triphosphate. And then the magnesium ions are also there to help to spatialize the negative charges of the phosphate group, because there's three phosphates, guanosine triphosphate. So this is kind of just a brief summary of like the pathway it takes. So as we can see, when light hits your eyes, you know, CGMP gets cleaved into GMP, so that goes away, and that in case causes calciums not to be able to come into the cell, because the channel will close due to the lack of CGMP. So this in turn activates the GCAP The lower calcium levels activates the GCAP, and then the GCAP is what binds to the RET GC1, which in turn converts here GTP into cGMP, and then allows for calcium to flush in. And once the calcium flushes in, it kind of tells it to, stop producing cGMP because we have enough calciums inside the cell. And we're going to talk a little more about the GCAP when we get to that. So outside the retina, it is a homodimer. And as we know, homodimer means it has one subunit and it has two main structural domains. It has the extracellular domain, which we're not going to focus on. We're going to focus more about the intracellular domain. So in the extracellular domain is basically just whatever's on the outside of the membrane. And then the intracellular is what's inside the membrane. So it's a type of I'd say like integral protein that we talked in class where it spans the entire membrane. So we have two states, kind of similar to how hemoglobin works. We have the active R state and then we have the inactive T state. In the R state, as we said, is the active state. So there's low CA2 levels. So that in turn activates GCAP. GCAP activates the RET GC1, which in turn flushes the calcium in. So the inactive T state, so when it's not working, it's basically when there's already a lot of calcium inside the cell, so it doesn't need to open that domain. So that's why we call it the inactive state. And then just some fun facts as well, nothing particular here, just the intracellular domain. We have the different functions that kind of help convert GTP into cGMP. So we have the kinase homology domain, which is the addition of phosphate groups. And then we have the catalytic domain, which is kind of the binding site. And then we have the dimerization domain, which basically in its active state, so where it needs to be open, is homodimer. But in order for it to be an enzyme, it needs to be a dimer. So it undergoes dimerization. This is kind of just a comparison between the two states. As we can see in the active state, the GCAP is binded and then the domain is closed right here. So it needs to be active in order to convert the GTP here to the cGMP so it can become open. So once it's open, it doesn't need to be working anymore. So it kind of just sits there, which is also called the inactive state or the T state. So basically the mechanism, the light hits the retina and causes a decrease in cGMP. So this closes that membrane we're talking about. So the membrane we were talking about is the cyclic nucleotide gated channels, and that in turn lowers calcium levels because calcium cannot go inside the cell. So that low calcium level allows GCAP to bind to RET-GC, and that in turn activates the enzyme. So the two catalytic subunits, so the dimer we were talking about, they're complement, so they bind to two magnesium ions and a GTP molecule. So the activated RET-GC1 catalyzes the GTP to cGMP, so that gets rid of a phosphate group and then cyclizes the GTP molecule. So the rise of, the cGMP that we just produced opened up the cyclic nucleotide gated channels, the CNG we see here, and that in turn allows calcium ions to rush in, or the influx of it, and that helps recover vision. So basically you can kind of stare at the sun again, but not advised. So this is just another image about how the entire process works. As you can see here, so the light decreases cGMP levels, channel closes, calcium level goes down, lower calcium level activates GCAP1, which binds to RET-GC1. RET-GC1 goes under changes, converts GTP to cGMP. And this is just another image right here. So let's talk a little more about the different ions and then a little more about GCAP and what that does. So during the dark stages, so when you're not, actively using that photo receptor, there's high amounts of calcium. So that's when it's inactive. So it inhibits it from creating cGMP from GTP. We don't need RET-GC to be active during the dark stage. So once the light, however, hits our retina, there in turn is a low amount of calcium, which because there's a low amount of cGMP, so this in turn activates the RET-GC1, which produces a cGMP. So that ultimately opens up the CNG, which is that membrane channel, the cyclic nucleotide membrane channel. So sometimes GCAP will also bind to magnesium ions when there's no calcium, but that is in the active stage because there's no calcium. So it kind of signals it to, okay, I need to bind to the red GC. So there's also other involvements that CGMP. I mean, I'm sure you guys have heard of GTP before, but this is just how it works in the cyclase guanylase 2D gene, which codes specifically for the retinol side of it. But CGMP, it is a common secondary messenger. As we talked about, the phototransduction pathway, it regulates the CNGs and it initiates signal transmissions, activations of protein kinases, and then other pathways that require, a messenger to go intracellularly. So things that cannot, easily diffuse into our cell that requires, additional signal. So basically, just a quick summary. GUCY2D is the gene that makes the ret GC, which is that enzyme protein that we were talking about. It catalyzes the conversion from GTP to CGMP. So in order for GCAP to bind to ret GC1, there needs to be a low calcium level, and that happens when light hits the retina. And then ultimately, ret GC1 converts GTP to CGMP. This cascade opens up the cyclic nucleotide gated channels, and that allows the inflow of calcium, and then for the cascade to happen again. So the two questions I have here is, how does GCAP and calcium levels affect and regulate the GUCY2D during the phototransduction cycle that we were talking about? And the second one is, what channel is activated by the ret GC1? And then what type of channel is it? And does the channel require ATP? Thank you guys. I hope you guys have a good Easter.